Much of our ability to understand the very complex factors that impact protein evolution is dependent on the amount of data that we have available to us. And we really are in the middle of this massive change in how we do biology. And this is all due to big data, biotechnology, the ability to sequence vast amounts of data. And when we've got those big data sets, we can do things like make inferences, go from the general to the more specific, understand patterns that might be difficult to see on a much smaller scale. So we really are, this is a little bit dated. This was done, I think, about 3 years ago. And really we've come closer to this red line here, meaning that about every several months, 7 months, the amount of DNA data that we have available to us doubles. So realize we had in the late about 2007, 2008, noticed this dramatic increase in the amount of genetic data available to us. This really is what we call next generation sequencing, or big data available to us. And because of that, many of these patterns that we're talking about are beginning to be understood at better and better levels, and we're going to be able to do more and more with them. So just be aware that our ability to sequence DNA is dramatically speeding up. If you notice the scale on this graph, this is a logarithmic scale. So every time you go up, you triple the amount of data. So this is 1 terabyte, which is a trillion. All right, that's one more than a gigabyte. So this is tera base pairs. A trillion. This is peta, so a zillion. I forget what the next one is after a zillion, and then all the way up. A zettabyte, I don't even remember what that one is. But anyway, every time up, so this is 1,000 times as much as there below. That's 1,000,000 times there below, and this is 1,000,000,000 times as much information as we are right there. So that basically right there is 1,000,000,000 billions. So the short take-home message is that our ability to sequence DNA and the amount we have available to analyze is just increasing exponentially, very, very rapidly. And so that has allowed us to say a lot and begin to analyze data at a very large level. Now, one of the things that we've been able to gather more information about is the mutation rate and the substitution rate that we have at the DNA level, and then how that leads to differences in the proteins. So let's define these two and make a distinction between them. The mutation, well, before we do that, let's define rate, right? A rate is always some measurement over some period of time. So your rate of travel is miles per hour, right? Or kilometers per second, or meters per second, whatever you want to do it. So realize that when we're talking about a mutation rate, we're not only talking about amount of change that's occurring, but we're talking also over some measurable period of time. And for biological measurements, we will often use per generation, right, or per cell cycle, but per generation is a very, very common measurement that's used. And so mutation rate is the number of mutations that accumulate per generation. Okay, so the number of changes in a genome over a single generation. Okay, so that is known as the mutation rate. Now, if there are no evolutionary forces acting on a sequence of DNA, then the mutation rate and the substitution rate are the same. But the substitution rate is the number of those new mutations that come into the population that become fixed and become part of the genetic history of that entire population. So fixed, remember, does not mean repaired. It's sometimes a common misinterpretation of that word. So maybe fixated is a better term. So the number of mutations that become fixated or go to 100% in the population, that's the substitution rate. And again, these numbers are equal if there is no other evolutionary force other than mutation acting on a population. Well, genetic drift always acts to some extent, but that's offset by population size. So the short answer is the mutation rate is equal to the substitution rate unless there is natural selection. But when there is natural selection, then suddenly the substitution rate becomes very different from the mutation rate. We might have all sorts of mutations occurring, but if those are removed by natural selection, or if some of them are driven forward because they're good by natural selection, then our substitution rate, maybe even for different parts of the genome, will be different than the overall mutation rate. Now, the mutation rate itself is subject to variation. It's highest in viruses, including RNA viruses, HIV, these little viroids have very, very high mutation rates. Viruses in general have high mutation rates. Bacteria have somewhat elevated mutation rates, and the mutation rates generally are lower. Now, this is also corrected for genome size, because these have little tiny genomes, but even when we correct for that, we still have a much, much higher mutation rate in HIV, for instance, than we do in any eukaryotic organism. And these are adapted. The high mutation rate is a good thing for a virus, where that same mutation rate in a eukaryote would have a very detrimental effect. So the main thing to know is that we have a very wide range of mutation rates across different organisms. Okay? Not only that, but even if we look within the human genome, so this is looking at just one single genome, each number here represents a human chromosome. And then we have an average rate of mutation within that chromosome and a range. So chromosome number 1 has, an average medium to high rate of mutation and not a lot of variance. But chromosome number 9 has this huge amount of variance and an overall elevated rate of mutation. So even within a same genome, we can get very different rates of mutation. And mutation rates are impacted by many, many different factors. So let's just talk about 2 of them, just to give you kind of a foundation. Number 1 is the mistake rate, right? How often polymerase makes a mistake. And that's actually one of the main reasons for why viruses have a higher mutation rate than eukaryotes, is their polymerases are messier. They make more mistakes when they're copying DNA, and that's an adaptation for viruses. It's a good thing for them. Whereas the polymerases that copy eukaryotic DNA have a much higher fidelity. They make many fewer mistakes. And then the second thing that we might want to consider, main thing, there are other minor things also. The second main thing when we're looking at mutation rates is how often those mistakes that the polymerase makes are caught by repair mechanisms, because there are repair mechanisms and these are more efficient in some organisms than the other. And so we might have different rates of mutation mistakes that are made here. We might have different repair mechanisms, although there could be many other factors that are also influencing these overall mutation rates. Now, recombination plays a major role in eukaryotes. Recombination includes both independent assortment of chromosomes, but also, and maybe more importantly for our purposes, it includes this crossing over. And the reason that recombination is important is if recombination is very frequent if it happens commonly, then we can treat the individual genes as if they are units unto themselves, and they won't be impacted by alleles next to them. However, if recombination is rare, then there is an evolutionary force that is imposed by the alleles that are next to a gene of interest. So if that's not entirely 100% clear to you right now, that's fine. It's kind of going back to things you should have learned in Bio 1 and maybe talked a little bit about in the higher division courses. But just be aware that high rates of recombination basically mean that we can treat a single gene as an independent unit, and it's not going to be impacted by the alleles that we find next to it. Now, when we look at proteins, we notice a number of patterns, and we're going to talk about 2 very, very general patterns. The first we will call purifying selection. Sometimes it's called negative selection, so I put that in parentheses, but we'll know it as purifying selection. And this is an example of how the mutation rate and the substitution rate can be different. So mutation, at least point mutations, which we're going to be the main source for variation we're going to be looking at, those point mutations are effectively random. They occur equally across all parts of the genome, and if we're looking at a gene, there's going to be no pattern at all with regards to first, second, or third position. However, we notice in many, many genes that when we compare sequences between species, that there is a very, very clear pattern. And I review this when we talk about this to some extent in evolution class. So let's look at this pattern here. So this is in reading frame. This is an insulin gene for 4 species of mammal: humans, dogs, bats, and whales. Here's human insulin. Notice that there's a very clear pattern. That's the first, that's the second, and that's the third position in the codon. Look, there's a mutation at the third position in the codon. The next codon has no mutations. The next codon has no mutations. The next codon, oh look, there's a mutation at the first position. But after that, 1, 2, 3, 1, 2, 3, 1. So in this region, we do seem to see mutations more commonly in the first position. But if you look farther on, 1, 2, 3, mutation, 1, 2, 3, there's a mutation, 1, 2, 3, 1, 2, 3, mutation, 1, 2, 3, mutation, 1, 2, 3, 1, 2, 3. So again, we go over and over. But most of the mutations at the end of this gene are only visible at the third mutation, at the third position in the reading frame. Now, we could make the inference that, well, maybe for some reason there's some mechanism that causes mutations to occur more frequently at the third position. But that doesn't seem to be backed up by the data. In fact, what's going on is mutations are occurring equally, but mutations at the second and first position in the reading frame are more commonly removed by natural selection because those make changes to the amino acid. And so what this ends up resulting in is more mutations for many proteins. More mutations will accumulate at the third position. And so when I look, and it's especially apparent here at the end of this alignment, this is the amino acid translation of this nucleotide alignment. And notice that at the end especially, we have very, very few changes. But even here, we have many changes that do not result in an amino acid change. So purifying selection is removing most of the mutations that make an amino acid change, but allowing many of the mutations that are synonymous, that don't change the amino acid, allowing many of them to remain. And so this is one of the factors that can impact protein evolution. Now, the opposite of purifying selection is positive selection. And positive selection is when there are actually changes to the amino acid. Those changes confer a benefit on the organism. And so positive selection causes different genes to diverge in function. So if I take insulin from many mammals, and it's not 100%, but many mammals, and put it into a human, it still will function as human insulin because it's been under strong purifying selection. However, there are many other proteins where that wouldn't work. If I took a certain homologous protein, and I can identify it based on structure or based on overall sequence similarity. But if I took it out of a dog and put it into a human, for many proteins, they would not be compatible. And that's because we've had positive selection that has caused those proteins to diverge. Now, we can even look at within a single protein. This is a diagram where what they have done is they have looked across many different homologous proteins in different species, and they have mapped the amount of diversity and change that has occurred. And some areas seem to be very, very conserved, and other areas seem to be able to change and mutate and accumulate mutations much more freely. So we might even see within a single gene an area that is under purifying selection and an area then that is under positive selection and takes on a new function. So for instance, if we're looking at, say, a membrane protein, the part that allows it to stick in the membrane might be very, very conserved. But if we're looking at maybe a receptor outside of that on the same protein, it might be subject to positive selection and might change very, very rapidly. And so we can make these maps and start to investigate patterns across different proteins. And so this is known as the fitness density, where organism or parts of a protein that change very, very little have a high fitness density, because any changes at all that make a change in the corresponding amino acid will most often be removed by natural selection and therefore stay very, very conserved. So these blue areas have a very high fitness density, whereas the most flexible and the most changeable ones would have a very low fitness density. They can change quite a bit, and it has very little impact on the fitness of that protein. And so we can begin to put these changes into their three-dimensional protein structure. And remember, protein structure, the shape of the protein, is very, very indicative, and the structure completely has an impact on the function of that protein. Now, there are some other factors that need to be considered, and hopefully you're getting this idea that protein evolution is very, very complex, and we're really only beginning to scratch the surface of our understanding of what's going on. So another thing that can impact the mutability or the flexibility of a protein to evolve is its role in genetic networks. So if a gene has a role where it interacts with 4 different downstream or maybe upstream, it really doesn't matter, but if it interacts with 4 different proteins, that's going to put a high level of constraint on that gene, because if it mutates to have a better interaction with one of those, how is that going to impact its 3 other jobs or its 3 other interactions? Whereas if we have a gene that only has a single or maybe 2 interactions, right, like PLK3, it might be a little bit more flexible and can explore and mutate and change a little bit more. So gene networks can have an impact on mutability. And this is just one aspect of pleiotropy. So review hopefully, but if not, learn this vocabulary term. Pleiotropy is where a single gene has 2 or more jobs. And so the more pleiotropic a gene is, the more constrained its evolution will be, because it won't be able to, it just has more interactions and more things that it has to do. So there's a higher probability that a mutation in that would not be a good thing. Okay, the second factor is developmental timing. So if a gene is impacting early fundamental development, it might be much more constrained and much less able to evolve than a gene that is only involved in very, very late development. So those are probably going to be a little bit more flexible, and we might see more changes occurring there. So developmental timing can play a role in the variability of genes that are involved in that development. And then finally, another very important one that we have noted from all of this data are expression levels. So this is a heat map and is just there for visual purposes, nothing that you need to gain out of this diagram. But it shows different expression levels in different tissues. We could even do expression levels across different species. Okay. Now realize that, and so basically, the higher a gene's expression is, the more constrained it is on average. If a gene is expressed only in one tissue or only at very, very low levels, then it seems to be more free to evolve and change. Now realize there are confounding things going on here. So for instance, a gene that is expressed in many, many tissues typically also has higher overall expression levels. And so those two things might be confounding. And so these things really are related. So we've talked about mutation rate and recombination rate. We've talked about then all of these things can interact at one level or another. The number of protein interactions in a network, whether a gene is really critical for function or maybe it's not that important for survival. So all of these things interact with each other. Sometimes they are correlated or inversely correlated, and all of those have an impact on protein evolution. And so you should understand these concepts generally and this complexity. Now, because of all this complexity, we are still unraveling all of the things that are going on, and we'll look at some analytical methods in later discussions. But for now, I just want to lay the groundwork, the conceptual groundwork for understanding protein evolution. Now, there are some other things that I want to basically just define, and for now, just know that these play a role, some of which we have mentioned before. One of these is both gene duplication and motif duplication. So if we have a gene that's duplicated, so I have 2 copies, that can release some of the constraint on that gene, so it's more free, or at least one of the copies is more free to evolve and adapt because the other copy can maintain the ancestral conserved function. Now, a motif is simply a region of a gene. It might be a domain or it might be several domains together, so it's a little bit more of a flexible term. But a motif means when we duplicate and get extra copies of that region of the protein. And so again, that can release one of those regions from the constraint and allow a little bit more flexibility in evolution. Domain shuffling is the movement of one region, functional region of a protein to a completely different protein. And so in a way, we can kind of mix and match, and this is a form of a mutation, but we can mix and match different proteins and end up with potentially different outcomes. Like any mutation, when these occur, there's no a priori prediction about whether it will be positive, negative, or neutral. In fact, probably most domain shuffles are negative and are removed by natural selection. But every once in a while, we get a new gene combination that's part gene A and part gene B, has a brand new function and does something novel. And then finally, another thing we may need to consider is horizontal gene transfer. This is particularly important in bacteria and other prokaryotes, but plays a role also, although a minor role, in eukaryotes. So if you don't know that, look it up. Well, let's give you a quick definition. Horizontal gene transfer is when a bit of genetic material is transferred from one species to a completely unrelated species. And so after that transfer, the recipient of that DNA has a little tiny bit of DNA from an organism that is not its direct parent. Okay, and again, this is rare in eukaryotes, although it does happen, but is very, very common in bacteria via plasmids. And so if a gene is transferred from one species into another, that's going to impact its evolution and the ability of that protein to change and